CRISPR was supposed to set us free.
A master key to evolution's locked, and instead it chained us tighter. Your DNA isn't yours, it's leased. Your lungs, your blood, your eyes. Premium software billed monthly. You miss a payment, well, the corpo tower decides if your heart keeps beating, eyes keep seeing. But it won't be that painless. Cells folding wrong, muscles spasming like corrupted code, tumors blooming where the edits once lived, the billboards promised salvation. But the reality's uglier. Blue-collar workers with half-working mods, kids spliced on credit they'll never repay, bodies glitching when the subscription lapses. CRISPR wasn't liberation. It was privatization of the human genome. And in this city, your DNA isn't you. It's property.
Most of you have heard of CRISPR, but where did it come from and who's behind it? And who do we blame when we find out all the cryptids are just botched genetic experiments? Let's rewind and see how humans learned to hack DNA safely. Well, for now. In the early 1970s, Hamilton O. Smith, Daniel Nathans, and Werner Arbor noticed something weird. Some bacteria resisted viruses that easily infected others. They called this host-controlled restriction and modification. Think of it like this. Jason Voorhees goes to a summer camp and does what Jason Voorhees does best. But when he tries to summer camp across the lake, all the kids are homicidal and have shotguns and flamethrowers.
Bring him the blood of the Outlander!
Also, can we all agree that we're new here? Science is really cool, but I'm obligated to ask dumb questions because I myself identify as a doofus. What is a virus and a bacteria?
Black goes on the red with the, if we positive.
Bacteria are microscopic single-celled organisms. They're simple, they're everywhere, they multiply in two, which is called binary fission, and they are what get you sick and also keep your gut healthy. Unless you pound energy drinks, then you're a bacteria murderer. Simple enough. A virus is even smaller than bacteria. They are parasitic and require a host to live. That host is your cells, which it hijacks and forces them to make more viruses instead of doing what they were supposed to be doing. Like if you're replacing an alternator and your boss walks up to you and says, "Hey, I know you're busy, but the parts room needs organized. So how about you grab a couple other mechanics and do that? I'm gonna go get a burger." Your boss is a virus.
No, it's great. I love working at Dunder Mifflin.
Back to it, Werner Arbor hypothesized that bacteria either had enzymes to cut up invading DNA or modified it to protect their own. Studying E. coli, he found viral DNA cut into fragments, proof that DNA cutting enzymes existed. He called them restriction enzymes, though personally, I'd have called them samurai or cleaver enzymes. I remember playing Fable 3 and being a chubby horn dude that dual wields cleavers and just chops people up, and I like to imagine these enzymes as that.
Shut your preaching ass up. Nobody care about that shit.
Hamilton Smith and his team at Johns Hopkins isolated the first type 2 restriction enzyme, HindD2. HindD1 was sloppy, like swinging a machete through overgrown brush. But HindD2 is a scalpel. It cut DNA in the exact same spot every time. After HindD2 is isolated, Nathan Daniels proved its capabilities by using it to cut simian virus 40 DNA, which is a small DNA tumor virus that infects monkeys. This experiment had HindD2 cut the virus at 11 sites, making consistent, predictable pieces, and then use these pieces to map the entire genome of the SV40 virus, which is the first time this was done, and proved that HindD2 could be used as a precision tool in genetics, which earned all three of these nerds the Nobel Peace Prize in 1978.
Well, let's get those nerds! Nerds! No!
Round of applause for the men that will be eventually responsible for the real man-bear pig. This is why there's cryptids. They're just botched genetic experiments that got tossed into the North Sea.
Get rid of that thing, will ya? Son?
Alright, so around the same time, another scientist, Herbert Boyer, discovered another restrictive enzyme in E. coli, naming it EcoR1. This enzyme was different. It didn't cut in a straight line. It was staggered, leaving what they called sticky ends, which made splicing in foreign DNA possible. And just 3 years later, that's what happened. But before that, dumb question, difference between DNA and plasmids. Not sure, but it's important in a minute. DNA, deoxyribonucleic acid, the molecule that carries genetic information. Your blueprint, your main character build, necromancer, knight, mage, berserker. Plasmids are called bonus genes, only found in bacteria, archaea, and eukaryotes. I don't know what those other two are, so they're not real probably. Plasmids are located separate of the main DNA but act as buffs, like a specific antibiotic resistance. So it's basically your skill tree in Fallout. Want to have radiation resistance? Pick rad resist. You want to do more damage to female enemies? Pick lady killer for some reason, weirdo.
You're gonna hit a woman? Get out of here!
Now that that's out of the way, Herbert Boyer, expert in restrictive enzymes, and Stanley Cohen, expert in plasmids and bacteria conjugation, performed a couple of experiments. During the first, they isolated a plasmid from E. coli and used the EcoR1 enzyme to cut it open. They then inserted a second plasmid carrying a different antibiotic resistance. The result? The E. coli now carried a single recombinant plasmid containing two different antibiotic-resistant genes, allowing the bacteria to survive on media containing both antibiotics. The second experiment a year later seemed to be just for funsies. They spliced E. coli DNA with random DNA from the African clawed frog. I say random because they didn't have the technology to know exactly what parts of the DNA they were using. Also, let's be clear, one is a bacteria found in Dookie, the other is an amphibian. That's mad scientist stuff, but this really worked. The recombinant DNA took, survived, and replicated. And those replicated E. coli bacteria all held those fragments of frog DNA.
Like raw poop, human poop, raccoon poop.
Let's all take a moment to think about how terrifying this could get. Now, Plum Island. Just kidding. Maybe another time, but if you want me to get into conspiracies surrounding science and technology, I'd be happy to oblige. Okay, so in the 90s, W. French Anderson and his team at NIH performed the first approved human gene therapy on a 4-year-old girl named Ashante Da Silva. We'll get into that, but in between discovery and application, some important milestones happened. So let's knock those out. 1975, the famous Asilomar Conference was conducted and set guidelines to recombinant DNA because, of course, modifying genes in DNA is kind of crazy. In 1977, Genentech, a biotech company founded by Boyer, who we talked about earlier, produced the first synthetic insulin gene and inserted that into a bacteria, and in 82, that insulin, called Humulin, was FDA approved and mass-produced for human medicine.
Diabetes!
Also in 1980, the US Supreme Court ruled that genetically modified organisms could be patented. Then in 1984, this is an important one, were the first reports of retroviral vectors, which is the use of a virus as a delivery system for genes. This works because we asked the dumb question, what is a virus? We know that in humans, viruses need to insert their genetic material into human cells to function. Because they can now modify the virus, they can take out what's bad, put in what's good. That virus will then insert itself into white blood cells, which will replicate and pass on the good genes. Fast forward to 1990, W. French Anderson performed the first approved human gene therapy on a 4-year-old Ashante Da Silva, who had ADA-SCID, aka bubble boy disease. They used the retrovirus to deliver a healthy ADA gene into her white blood cells, and it worked. She could leave isolation and live a more normal life, but it wasn't a cure. The white blood cells had a limited lifespan, so repeat treatments were needed. Even so, this marked the start of a promising future in gene editing. And although the tools so far weren't precise, you gotta start somewhere. Now, all this gene therapy eventually paved the way for gene editing. The first editor? Zinc finger nucleases. I also thought that was a weird name. A tool that fingers your DNA so good it can alter it. Sign me up, probably.
Wait a minute. Are you telling me I need a prostate exam?
Invented in the 1990s by Carlos Barbas III and Diana Carroll, who realized you could take a naturally occurring protein called a zinc finger and program it to recognize almost any DNA sequence you want. It's named that because it's stabilized by a zinc ion and looks a little bit like a curled finger. Gross, brother. So zinc finger proteins recognize 3 DNA letters at a time, and DNA uses 4, which represent 4 different chemical bases that ends up being the instruction manual for life. So it works like this. DNA is the book of life. The zinc finger can only grab 3 letters at a time, which by itself is useless for editing, but they found if you string enough of these together, they can grab onto a specific word in the book that only appears one place. Now, when they pair these fingers with a restrictive enzyme, in this case FOC1, which is a restrictive enzyme found in a marine bacteria, so it feeds exclusively on crayons.
That's a war face! Now let me see your war face!
Just kidding. Bonded with this cutting enzyme, it can remove that single word and replace it with a completely different word, changing the meaning of that sentence forever. So instead of this sentence saying, I love cupcakes, it would say I fornicate cupcakes. Same, but different.
James Bond would love this cupcake.
You get the gist. ZFNs were a giant leap forward in the gene editing lore. Powerful and precise in use, but complicated and expensive to make. But in 2010, that would all change. Transcription activator-like effector nucleuses, or TALENs for short, there's the cool name we were all looking for. These were developed after scientists studying a plant pathogen called Xanthomyces. Dumb question time. A pathogen is any microorganism that can cause disease, like a bacteria or a virus. In this pathogen, they found tails, transcription activator-like effectors that were precisely binding to the plant's DNA to turn the genes on or off. The researchers then thought, if we pair this with that FOLK1 cutting enzyme, I wonder how precise this could be for gene editing. The result? A 1-to-1 code. ZFNs were limited to triplets, 3 letters of matching code at a time. A TALEN could program a single letter at a time, making them completely customizable and less likely to cut in the wrong place, which would be an issue.
I made a mistake. A terrible mistake.
What this did was create a clean 1-to-1, much easier to program and precise gene editing tool that was eventually used in zebrafish, rats, plants, and human stem cells, opening the door and expanding genetic therapeutic research, leading us to our current main character, CRISPR.
Is the sophisticated gene editing tool known as CRISPR.
CRISPR, the acronym for Clustered Regularly Interspaced Short Palindromic Repeats. I don't know why my voice went country, it just felt right in my soul, which describes what researchers have found and recorded between the early 80s and 2000s, but had no idea what they were. Let me explain. Early on, researchers from Osaka, Japan found short repeats of DNA in E. coli separated by little spacers. No idea what they did. Little did they know, the most advanced gene editing tool was right in front of their faces. Later on in the 90s and early 2000s, the same DNA repeats were found in different microbes, showing that this was not specific to one bacteria. And around the same time, scientists in the Netherlands proposed the acronym CRISPR to describe these unique repeats. These same researchers found nearby genes that seemed to pair with the presence of CRISPR. Wherever they found CRISPR, they found these genes as well and named them Cas for CRISPR associated. Now the breakthrough. In 2005, multiple independent research groups identified that these spacers they were seeing actually matched viral DNA. What this meant was that these bacteria were taking mug shots of past viral infections for future defense, and this Cas protein was doing the cutting, securing these mug shots. It was the restrictive enzyme, and the link showed this was the bacteria's immune system. And the proof came in 2007 when the company Danisco ran an experiment that showed when bacteria survived an infection, new spacers of viral DNA were added to the CRISPR system, which then made that bacteria resistant to the virus. So how did this translate to editing, right? Well, in 2012, Emmanuel Charpentier and Jennifer Doudna found out that Cas9 used a piece of RNA provided by the bacteria that acted as a guide to recognize and attack previous viral infections. First, yes, the last 3 letters of Jennifer's name are DNA. Second, there ended up being multiple variations of the Cas gene. In this case, Cas9 was used. Simply put, the bacteria DNA is a car and it uses GPS to guide itself to a previously programmed destination. The GPS is the RNA. A lot of acronyms. Reminds me of the Army. Now these borderline mad scientists thought, you know, if we pair this up with our own RNA and gave it to Cas9, we might be able to get it to do whatever we want. Mad scientist Fan Zhang used Cas9 in a human kidney cell essentially to test its precision, which showed just how precise it could be. Then turning that to show how well it could target the CCR5 gene, which is what HIV uses as a host cell, hoping to make humans resistant to HIV. Take that, CIA. Douchebags.
The CIA did what the CIA does.
Then George Church's team wanted to see if they could use Cas9 to turn genes on and off without cutting them. So they created a dead Cas9, paired it with RNA instructions and proteins that turn genes on and off. And they did. They activated and repressed genes in human cells without altering the actual sequence, proving CRISPR could be used as a gene regulation tool as well as a gene cutting tool. Honestly, mind-blowing. Since this, it's been full throttle. Therapies for sickle cell, lowering LDL cholesterol. Yale is using CRISPR to modify combinations of genes at once in rats to study its effects on cancer, immune responses, and to find out how diseases actually happen. It's being used in agriculture as diagnostics for crops for early disease detection that can be seen by the farmer almost immediately after infection to help reduce crop loss. Genome-wide CRISPR screens to manipulate thousands of genes in parallel to see which ones are responsible for certain traits or sicknesses, and editing current toxic bacteria, like E. coli, to reverse antibiotic resistance so they become easier and more effective to treat. Now, this is really cool, right? But it's early and there are ethical concerns. Mistakes are still being made, like off-target editing. In 2018, an institute in the UK saw CRISPR delete huge chunks of DNA trying to repair a cut it had made, which could lead to cancers and disorders that don't exist yet. In the US and Sweden, they found that CRISPR sometimes killed off healthy tumor-suppressing genes, leaving only the faulty ones, promoting tumor and cancer growth instead of suppressing it. So basically what happens is CRISPR relies on a 20-letter RNA GPS guide to find the DNA scientists want to cut, right? But if any DNA has a similar guide, it could cut there too. So flip a coin. Aggressive unknown cancer on one side or chromosome disorder no one's ever seen on the other. But hey, our baby needs blonde hair, blue eyes, be 6'4", have an IQ of 150, and be named Tanner for some reason. CRISPR could be a miracle tool or an absolute disaster. But either way, it's not slowing down anytime soon. And that's the ARC. A weird little bacteria defense system turned into CRISPR, now rewriting everything from medicine to agriculture. Today it's curing a disease and making crops more resilient, and tomorrow it might mean picking your child's height, eye color, or IQ, or worst case, unleashing mistakes we can't put back in the box. Gene editing is powerful, it's exciting, and yeah, it's also a little terrifying. The future of DNA isn't some distant sci-fi. It's already on the operating table. The only question left is, do we control the code? Or do the people that will control the code control us? And that's all I have. Thank you for listening. Hope you come back for more.